Growing microbes might be compared to a gardener trying to get a rare orchid from the heart of the rainforest to flourish in a greenhouse.
If you look carefully, you find the requirements. And it's very similar with the orchid. Some you have to hang them up into the roof, otherwise they will not be happy. Some people say I have a green thumb, but you see it's not so green.
In the lab, Stetter has reached the moment of truth. Has his green thumb produced a happy, multiplying population of Thermatoga? Or is something wrong with his recipe and the microbes have died?
Oh, it looks excellent. Before it was completely clear, but now the microorganisms have been grown and there are about 100 millions per cubic centimeter. And so, therefore, it's turbid and not clear anymore. And that we know it was growing very well.
By morning, the tiny sample of Thermatoga from Volcano has grown into trillions of cells of Thermatoga, which have been collected on this special filter.
Well, that's a lot. That's about 10,000 billions of cells. And they're all alive. And this is the real color of this stuff. It's almost like yeast.
Now, Stetter and his team have grown more than enough Thermatoga cells, enabling them to extract more than enough DNA for scientists to work with.
That's DNA from Thermatoga maritima, and that's a lot.
It is gooey material like this that contains the instructions for creating all life on Earth.
Normally, we work hard, but this evening, let's drink hard. Prost! Cheers!
Carl Stetter's harvesting of Thermatoga's DNA is a remarkable accomplishment. But for other scientists, harvesting the DNA of an organism is not an end, but a beginning.
Damn good stuff.
In the 1960s, most scientists believed that creating a new tree of life by comparing the DNA of different species was impossible. But in Urbana, Illinois, Carl Woese, a jazz aficionado and molecular biologist, disagreed.
You have to hear the tune, as it were. You got to have an ear. And you got to pick up on these faint tunes. And if you like them, you go where you can hear them better. You have to have your own particular sensitivity to the world. And there has to be parts of it that are beautiful to you because they're beautiful to you regardless of what anybody else ever thinks. And you see this all the time in an artist. And you see it also in good scientists.
For Woese, creating a tree that showed how life on Earth evolved, from the first form of life to the last, became an obsession.
I knew that this was a jigsaw puzzle that had to be put together. Now, in my case, I emphasize had to be, because I understood that to really be a biologist, to really understand biology, you had to understand where everything came from.
Woese focused first on the evolution of the first forms of life, microbes. The problem that had stumped everyone else was that, with microbes, you can't tell much about how closely they're related by just looking at them. But Carl Woese had an ace in the hole. A special chain of genetic material, special because it exists in very similar form inside every living thing. Woese began by cutting one of these special chains into sections short enough to manage. Next, he took pictures of the sections using X-ray film. Then Woese began the incredibly challenging task of figuring out in what sequence the four chemicals called A, C, G, and U were ordered in each section of the chain. Why go to such extraordinary lengths? Because Woese believed that this special chain could reveal how life on Earth evolved. He knew that this chain has done its job so well that it has been passed down from the first forms of life to all life that followed. Every bacterium on Earth today has inherited it, and so have you. It was like discovering that all the music ever written has a few notes remaining from the first piece of music ever created. Woese realized that by comparing the thousands of A's, C's, G's, and U's in the chains of two different species, he would be able to see exactly how much of the score of this special chain they had in common. The more of the score any two species had in common, the more closely related Woese believed they had to be on the tree of life.
And it was indeed just like putting a jigsaw puzzle together. You don't know at all what the picture is, and then you put all these little islands together and you say, oh, here's a part of it.
Fellow molecular biologist Norm Pace worked up the hall from Woese during the years he was attempting to create a new tree of life.
I think the remarkable thing about the Woese endeavor was that it was essentially single-handed. When you go into any high-power lab today, what you see is many people bustling around. And it was never like that in Woese's lab. It was always just Woese.
You got up in the morning, ate breakfast, and came into the lab. And then you put these silly X-rays up on your wall. And you looked intensely, but you had to be intense in order to get through this task. And this happened on Monday, Tuesday, Wednesday, Thursday, Friday, and week after week, and ultimately year after year. I would finish the day having looked at these films for many, many hours. And I would go home saying to myself, you know, you have destroyed your mind again today. And that's how it felt. Everything was dulled down. All my mental energy was used up.
I'm not sure that anybody except Carl Woese could have pulled off the articulation of the big tree. It required withdrawing himself, literally withdrawing himself from scientific society.
One key to Woese's approach was to search for the places where major new branches of life began, by looking for the place where a group of A's, C's, G's, and U's showed up in an order he'd never seen before. It was like only having heard notes arranged to play classical music your whole life, and then suddenly hearing them arranged in a rhythm and blues beat. And slowly but surely, one after another, the major branchings on the tree began to appear.
You could feel the thing growing. The islands became joined, being bigger islands, and then all of a sudden you see, this is what the picture is going to be about. And you gleefully throw in all the other pieces as fast as you can.
After more than 10 years, Carl Woese completed the puzzle, a new tree of life, created by comparing the genetic makeup of different species. When it comes to the branch of the tree that happens to include us human beings, Woese's tree points to a new, very different way of looking at ourselves. The new tree is based exclusively on how similar the genetic instructions of different species are. And the fact is that, just like the scores of country songs are remarkably similar to the scores of rock and roll songs, the scores of plants are stunningly similar to the scores of animals and human beings. And so the Wos tree, far from being just two branches divided between animals and plants, has only allotted animals and plants tiny twigs together out on the end of the branch we share with all other visible life.
It's difficult to see this tree here as your relative, because ever since recorded history almost, mankind has distinguished between animals and plants. These were the two great forms of life, and they were as different could be. But yet, scientists began to find out that there was a lot of commonality between plants and us, and that's the thing that has really sort of shocked even me, to find out not how different animals and plants are, but how much alike they are.
If you have a hard time believing that animals and plants are alike, consider what it takes to be different. When Wos decoded the scores of a newly discovered group of microbes, including a species that eats volcanic gases in the ocean, he found the letters arranged in patterns far, far different than any he'd seen before. It was as if Wos had discovered a new kind of music that human beings had never heard before. Wos found the same unusual pattern in some of the microbes that Carl Stetter had collected from hot springs. Wos realized that their unusual genetic material made these strange organisms so different from the bacterial branch and from our branch that they had to be given their own branch on the tree of life, which he named the Archaea, for ancient. Wos's work was especially meaningful to Carl Stetter, for it shows that the ancestors of all three branches of the tree lived in boiling water.
Due to the work done by Carl Wos, where these little tiny bugs are in the tree of life. And so it was so interesting, the hotter the temperature, the deeper the branches are, and this makes my work even more exciting.
For many scientists, Wos's tree has made the case for the scientific view of evolution more compelling than ever before.
It's my belief that Carl Wos has done more for biology than anyone since Darwin. Always before, evolution had been theories. Evolution had been notions, ideas, hypotheses. But as the molecular trees began to spin out, it was very clear that this was not theory. This was fact.
Ever since I was a kid, I've wanted not only to understand things superficially, but I wanted to understand them deeply. I need the truth. Some call this a search for God, and some don't. But you want to go deep, and you want to feel the truth. And priests do this, and many scientists do this.
Wos's work made it possible for the first time for scientists to systematically explore the invisible world that surrounds us. Now they could begin to ask, who lives here? Who are their relatives? And what are they doing? There was just one problem. Scientists could only ask those questions about the few microbes they had managed to grow in the lab and collect DNA from. Then Wos's colleague, Norm Pace, made a breakthrough of his own. At its heart was the new technology of cloning, which made it possible for Pace to take a single strand of microbe DNA and clone as many copies of it as he needed. Pace realized that he could now collect microbes from any environment and make copies of their DNA without ever having to grow them.
My feeling was that for the very first time, the door to the natural microbial world was open to us. That for the very first time, we could understand what the makeup of the microbial world is, and that is, after all, most of biology.
The first place Pace tried out the new technique was a hot spring in Yellowstone National Park. What Pace discovered staggered even him. Yellowstone's hot springs were home to not just a few otherworldly microbes, but to a stunningly complex ecosystem filled with hundreds of different species. And it isn't just hot springs that are teeming with microbes. It is every environment on Earth. Now scientists are reading the scores of thousands of previously unknown species of microbes, ushering in a golden age in the study of evolution and of what the tree of